Moje ime je Gregor Cuzak, danes sem vaš moderator in tukaj smo na dogodku, ki označuje pričetek delovanja Centra tehnologij celičnih in genskih terapij, pomembnega imenika v razvoju ne samo Kemijskega inštituta, ampak slovenske znanosti. Še večji pomen pa ima sam Center za razvoj medicine in dobrobiti pacientov. Pravzaprav v samem štartu delovanja, v samem pričetku prizadevanja za to, da pride do tega začetka tega projekta, je da vodilna misel, predvidevam tista, kar nas povezuje, recimo nas, jaz delam kot zastopnik pacientov in marsikdo tukaj še v občinstvu in tudi vseh raziskovalcev in ljudi, ki delate v farmaciji ali pa v zdravstvu, v medicini, ste zdravniki, to, kako pravzaprav koristiti pacientom v tej novi dobi, ki je pred nami in v dobi, ki nam odpira številne nove možnosti. So, dear ladies and gentlemen, my name is Gregor Cuzak. I am a patient representative. There are a few of us who are that, but I think everybody here is gathered on the basis of actually providing benefits to the patients in this new era of science. This is an important milestone for the Chemical Institute, but not just for Chemical Institute. Also for the wider community, for Slovenia, for research institutions, and not just Slovenia, but also Europe as a wider whole, including the partners within the project who are the building blocks of this project. So without further ado, I would like to invite the first welcome speech from Professor Dr. Gregor Anderluh, the Director of the National Institute of Chemistry.
It will not be a speech, really, and it will be in Slovenian. Drage dame in gospodje, spoštovani cenjeni gosti, predstavniki ministrstva, predstavniki akademskih inštitutov, predstavniki farmacevtske industrije, izjemno smo veseli in ponosni, da lahko danes štartamo na tak način ta izjemen projekt, ki smo ga uspeli z našimi raziskovalci tu na Kemijskem inštitutu in s pomočjo zunanjih sodelavcev dobiti v okviru evropskega financiranja. Kot je bilo že omenjeno, to je izjemen korak, ne samo za Kemijski inštitut, ampak tudi širše in to bi vam radi danes tukaj predstavili. Zelo lepo vas pozdravljam, upam, da bodo poudarki, ki bodo danes v tem prvem dnevu te otvoritve in začetka projekta bili koristni. Slišali bomo veliko nekih poudarkov o tem, kako je to organizirano v drugih državah in veseli me, da s tem pomembnim korakom bomo zdaj eno tako zelo napredno infrastrukturo tudi pri nas lahko vzpostavili en tak center. S tem centrom se Kemijski inštitut premika iz tistih osnovnih raziskav bliže uporabi. Tudi to je en od pomembnih korakov našega inštituta in zato je v naši strategiji to res zelo pomemben korak naprej. Ne moram in tega, da se ne bi zahvalil vsem vpletenim kot direktor inštituta, v prvi vrsti predvsem profesorju Jerali, njegovi ekipi, ki je poskrbela za to izjemno prijavo, ki smo jo na inštitutu pripravili s pomočjo projektne pisarne, ki jo je vodila takrat dr. Barbara Tišler, s pomočjo kolegic in kolegov tudi na Ministrstvu za visoko šolstvo, izobraževanje in šport, še takrat gospod Anjo Petra bi izpostavil tukaj, ki je zelo veliko pomagala pri prijavi. Sedaj pa seveda v naslednjem koraku pa tudi dr. Mojca Benčina, ki je pa stopila zdaj v tiste velike čevlje, ja, in bo zdaj ves projekt vodila in koordinirala. Tako da s temi, seveda tudi ta širša skupnost, ki nas je podprla zdravnike, tukaj iz Ljubljane, skupnosti pacientov, mednarodne partnere, to je res velik projekt, ki ga sami ne bi zmogli, in tukaj je res to nek tak, zdaj se ekosistem izoblikuje okoli tega in to upamo, da se bo tudi v prihodnje nadaljevalo na podoben način. Uživajte v današnjem dnevu, uživajte v predstavitvah.
I would like to invite dr. Valentina Prevolnik-Rupel, državna sekretarka. Najavljam dr. Valentino Prevolnik-Rupel, državna sekretarka na Ministrstvu za zdravje. Valentina, prosim.
Najlepša hvala vsem za besedo, hvala za vabilo. Spoštovani ladies and gentlemen, I'll talk in Slovene as well. Dovolite mi, da najprej izrazim res iskrene čestitke k temu izjemnemu uspehu cele ekipe Kemijskega inštituta za pridobitev tega res velikega projekta iz programa Obzorje. Gre seveda za pomemben razvojni projekt, ki je plod znanstvene odličnosti in inovacij ter sodelovanja med številnimi strokovnjaki. Tudi sama sem šele pred mesecem in pol prestopila iz raziskovalnih vod, delala sem na Inštitutu za ekonomska raziskovanja v državno pravo, tako da vem, kaj pomeni projekt iz programa Obzorje pridobiti in ga tudi voditi ter koordinirati, tako da vam želim vso srečo, dr. Benčina. To bo res veliko delo. Predstavljam si tudi vaše veselje ob današnjem pridobljenem projektu in res iskrene čestitke. V Centru odličnosti za tehnologijo genske in celične terapije bo potekal razvoj najsodobnejših in personaliziranih metod zdravljenja. Ta bo vključeval pripravo proizvodov genske in celične terapije za zdravljenje in njihov prenos v klinične raziskave. Tudi za Ministrstvo za zdravje je to vrstni razvoj ključnega pomena, saj le na tak način lahko oblikujemo modern, na pacienta osredotočen zdravstveni sistem. Če pogledate naše usmeritve zdravstvene politike za leti 2024 in 2025, ki smo jih ravno kar objavili, je to eden izmed naših ključnih usmeritev, se pravi na pacienta osredotočen zdravstveni sistem. Center odličnosti bo med drugim naslovil pomembno področje raka. V Sloveniji živi približno 120.000 oseb, ki so kadarkoli zboleli zaradi ene od oblik rakave bolezni. Rak ni ena sama bolezen, rakov je več kot 100 vrst in čeprav smo pri zdravljenju vedno boljši, je še vedno veliko vrst raka neozdravljivih ali težko ozdravljivih. Izjemen napredek na področju znanosti obeta, da bomo v prihodnjih letih znali pozdraviti vse več težko ozdravljivih rakavih bolezni. Prav napredni pristopi zdravljenja, kot sta genska in celična terapija, so izjemno učinkoviti zato, ker so ciljani. Prilagojeni so posameznim pacientom ali skupinam pacientov. V Centru odličnosti je individualni pristop do vsakega pacienta središčna točka pri razvoju novih terapij. V Centru odličnosti pa se ne bodo posvetili samo področju raka, ampak tudi drugemu področju, ki bo s pomočjo genske in celične terapije lahko učinkoviteje obravnavano. To je področje redkih bolezni, kjer bo glavna pozornost namenjena tehnologiji za pripravo naprednih zdravil za zdravljenje genskih bolezni, za katere še nimamo učinkovitih zdravil. Namen je seveda premostiti vrzel pri prenosu tehnologij iz predkliničnega testiranja v klinično fazo. Napredne tehnologije zdravljenja bodo v prihodnje lahko zdravile številne bolezni, ki imajo vzrok v genskem zapisu bolnikov. Center odličnosti bo prispeval k več kliničnim raziskavam in bo za slovenske paciente pomenil tudi boljšo dostopnost do najnovejših metod zdravljenja. To bo stičišče vrhunskih domačih in tujih strokovnjakov iz različnih disciplin, pacientov in njihovih organizacij. Torej še enkrat na koncu hvala vsem predanim in odličnim znanstvenikom, strokovnjakom, ki sodelujete v projektu. Prepričana sem, da bo zaradi raziskav v tem centru za številne paciente vzklilo novo upanje za uspešno zdravljenje njihovih težkih bolezni. Čestitke.
So the State Secretary emphasized the role of patients in the center, and this being a center of excellence for cell and gene therapies, it really is on the horizon of the Ministry of Health, which is really good news. What about the horizon of the Ministry of Higher Education, Science and Innovation? Vabim na oder dr. Jureta Gašpariča, državnega sekretarja iz Ministrstva za visoko šolstvo, znanost in inovacije. Ne vem, če sem prav povedal naziv ministrstva.
Dear Director, ladies and gentlemen, I have my speech, my best prepared in English, but I shall follow your example and your two and say only a few words in Slovenian. But you may use it then afterwards. Torej, drage kolegice, kolegi, ekipa Kemijskega inštituta, Gregor Roman, res v imenu ministrstva zelo vesel in ponosen, da sem danes tukaj. To je že, ali bom rekel, šele drugi tovrstni teaming projekt, ki ga v tej državi imamo in je res odraz izvrstnega sodelovanja in pa predvsem predanosti vseh, ki so bili v ta projekt vključeni. V prvi vrsti seveda bi podaril inštitut, v drugi vrsti moram podariti moje kolegice z ministrstva, takrat če MIZŠ za Ministrstvo za visoko šolstvo, znanost in inovacije, ki so ves čas stali v strani temu projektu. Zdaj, načeloma stališča ministrstva velikih pomislekov pri tem projektu ni bilo. Financer, vse tisti, ki delno plača račun, vedno ima pomisleke pri vsakem projektu, naj se že kako dobro sliši, ampak prav velikih pomislekov pri tem projektu ni bilo. Iz več razlogov. Najprej zaradi tega, ker je napisano tukaj, to mi je zelo všeč tukaj od spode kot moto tega projekta. Most med biomedicinskim raziskavam in njihovim prenosu do pacientov. Spravi zaradi tega, ker je zelo v ospredju transfer of knowledge. Po drugi strani tudi zaradi tega, ker gre za teaming inštrument, ki ga lahko s komplementarnimi viri financiranja izvrstno izkoristimo za to, da uresničujemo to, kar smo zapisali v vse naše strategije, v strategijo pametne specializacije, v strategijo raziskovalno-inovacijsko strategijo, v nacionalni načrt razvoja raziskovalnih infrastruktur. Se pravi, z združenimi močmi, z evropskimi in nacionalnimi viri lahko le dosežemo take prebojne rezultate. Država bo tukaj zagotavljala izgradnjo in nakup visokotehnološke specializirane opreme, ostale stroške pa obsoduje Evropa. Tako da še enkrat res smo veseli in ponosni in komaj čakamo, da bo projekt ugledal sadove, predvsem pa še enkrat in upam, da ne prevečkrat čestitke in predvsem uspešno vsem sodelujočim. Hvala.
I will use the same trick as before. I pick a word and then I try to build on it. The word was bridge. Right? So the bridge between the research and the application, some call it bench to bed. So it's really super important for us, patients, that what is discovered in the bios, on the bench, in the research really comes to the actual lives of patients, to the bed, where it's treated by doctors, and of course, the whole approval process, clinical trials and everything. I'm sure it's a delight and pleasure, as expressed by the State Secretary, to support such excellent projects as this one looks at this moment, but it shall prove its validity in the next years. Zdaj bi pa k besedi povabil doktorja Sama Zdera, vodjo kliničnega oddelka za hematologijo v kliničnem centru Ljubljana.
So, licensed to Slovenia.
Prosim.
Dobar dan, pravim. Tukaj sem že kar nekajkrat, tako da se počutim domače, tudi hlajene ste zrihta. Mi je pa vedno zadovoljstvo sem priti, ker vidim zelo veliko ljudi, ki jih res spoštujem, ki so mnogo pametnejši kot jaz, ki je znanost bila položena v zibko. Meni jaz, kar se znanosti tiče, ne bi bolj tako, ampak kar pa znam, je pa na preprost način to videti, kam to paše, tako delo. In kolega Seber, ki je vse to, kar jaz nisem, tako da se nekaj dopolnjujeva, in je opozoril to leta 17, 6 let nazaj. Od takrat naprej se trudimo za ta projekt in danes smo tukaj, ker mi vsi obetamo korist za naše bolnike. In to ni treba biti preveč pameten, zdravljenja, o katerih govorimo in ki bodo dostopna s tem projektom, ki se začenja, so danes stanejo za posameznika ranga pol milijona evrov. Mi imamo bolezni kokrčte in za vsako tako bolezen lahko najdemo 30-40 ljudi. Pa si izračunajte 10-krat 30 do 40 krat pol milijona evrov, pa pridemo na 200 milijonov brez večjih težav. Kdo bo to plačal? Ne vem. Zaenkrat nagrajajo zdravila, super, to že sludi nekaterih posameznikov. V Sloveniji imamo vse in še več. Ampak temu ne bo šlo tako naprej. In za znanjem, ki ga goji Kemijski inštitut s profesorjem Miralo na čelu, Gregorjem, si obetamo, da bomo lahko za ceno enega zdravili 3 bolnike. Naši izračuni kažejo v to smer. Kar pa pomeni, da bomo lahko našim ljudem ponudili zdravljenja, o katerih lahko sanjajo tudi v razvitih državah. Smo blizu začetne realizacije na enem drugem polju. Prihodnost je pa to, kar govorimo danes. In da ne bom predolg, vedno se potem zagovorim, sem daljši kot vsi ostali. Naj povem, da je danes tudi za nas to ogromen dan in da vidimo cilj pred sabo, ki se mu reče ponuditi neko povsem novo kompetentno storitev za naše državljane. In ne samo to, glede na to, kako smo zastavljeni, verjamem, da bomo postali regijski center za zdravljenje, za to vrstno zdravljenje. In bi se zahvalil vsem kolegom, s katerimi sodelujemo, v prvi vrsti vama, tudi Inštitutu za mikrobiologijo, medicinskih fakulteti, kliničnemu centru, Ministrstvo za zdravje, ker imamo zadnje čase podporo. In stvari grejo v pravo smer in ne moramo ne uspeti. Hvala.
Lahko še trenutek eno vprašanje, bom jaz postavil, pa bom potem vam še komentiral. Nazadnje, ko smo se srečali tukaj, ravno v tem prostoru, ste povedali, da za razvoj terapije, na kateri delate, pri kateri bo ta center, kot razumem, tudi sodeloval, da ste pripravljeni jeti na kosilo tudi s hudičem. Ali ste bili na tem kosilu s hudičem in kakšen je rezultat tega?
Dobro, to je tudi spodoba. Je pa res, da ko enkrat stvar je v denarju, jaz sem prvač konkreten, ne ovinkarim, stvar je v denarju in ko enkrat toliko denarja noter držaženega pride vse mogoče ljudi naokrog. Vse mogoče ljudi naokrog. Meni je v čast in ponos, da nihče v našem delovnem krožku ni vzel centa nikoli za svoj žep. Imamo čiste roke, stvari so pregledno narejene. In je pa tako, če hočeš napredovati, pa dobiti neke stvari, ki jih rabiš, se naučil pogovarjati se tudi z nekom, ki se 5 let prej ne bi pogovarjal nikoli nikdar. Zdaj mi ni problem, ker vem, da je v ozadju nek višji cilj. In mislim, da je to tudi prav način, ker ne nazadnje pa lahko nekoga, ki živimo namreč v svetu, kjer je denar glavna paradigma sveta vladar, vedno bolj, kar ni prav. In pozabljamo na tisto, kar delajo tukaj ljudje, na tisto človeško komponento, ki nam jo dokončno izbrisal ali pa zameglil še COVID na vse to. In dajmo spet postati ljudje. In mislim, da taki projekti ne samo, da nam se strani znanosti odpirajo vrata, ampak nam odpirajo vrata tudi nazaj v eno humano sfero.
Hvala. So, to summarize really briefly, Dr. Zver mentioned that the therapies that are to be developed here in today's world cost an immense amount of money. And there is a plethora of those diseases that need to be healed. And this would require an immense amount of money that is not readily available if we project forward. Now, with goodwill and a lot of humanity, transparency, which he has shown in the project that he is leading, I questioned him about whether he would be willing to meet the devil at lunch, which was his metaphor that he used. And he confirmed, and we see that he's progressing wonderfully on the CAR-T therapy. And it's my admiration and admiration of many people for what you are doing. Thank you very much also for the answer. Naslednji, če se ne motim, bo posnet, oziroma se javi preko telekonference. Ali je tu? So I shall still ask if Dr. Doru Leonard Irimije
was he ever seen?
I was asking whether you are here to connect via teleconference. So this is very immersive. Very immersive. Nice to meet you. Here is your microphone. Please introduce yourself and step in.
Thank you very much. Thank you very much for the invitation. Compared to the previous speakers, I do not have the luxury of speaking in Slovenia today, so I prepared for an English speech and I will continue like this. I also noticed that we are well ahead of time compared to the agenda, but I will definitely not speak for 35 minutes because I will have a like 35 minutes presentation tomorrow in the morning, when I will speak in depth about project implementation and reporting and expectation on behalf of the European Commission and Research Executive Agency. I would nevertheless like to point out a very few important things now. So first and foremost, congratulations for the success with launching this project. Also, thank you very much for the project team that was very cooperative and efficient during GAP stage. There has been a number of changes done for a number of reasons. We will develop on this tomorrow during GAP, so I'm glad that we completed this exercise in a very smooth fashion. So also, once again, congratulations for this, because with the move from Horizon 2020 to Horizon Europe, teaming instrument has become more competitive, and this is the third call organized for the teaming instrument, and this was the most competitive by far. So in 2 stages, call 115 proposal received. At stage 1, 32 were invited to submit a full proposal in stage 2, of which 12 have been selected to be funded. 4 additional ones have been included in the reserve list, of which 1 more in the meantime has also been invited to GAP. So there will be 13. Adding 13 to 10 remaining from the first call to 14 from the second call, that makes a very selective number of 37 teaming projects running now during the 3 calls. With regard to the teaming instrument, as you will know, widening program has been introduced first in Horizon 2020. That was in basically 8 years ago, has been introduced. And it was supposed to bridge the gap between lower performing and higher performing countries. And as you will know, teaming action is the flagship action of this program. Projects are generally large size. They are in the range of 15 million EU grant, to which at least 15 million complementary funding is expected. Even though it is a widening program, scientific excellence is still main expectation, but I would like to emphasize a few more specific aspects of the characteristic for the teaming projects that have been looked at very closely by the expert evaluators at the evaluation stage and will be looked at by the expert reviewers at each of the 4 reviews that we will most likely have on site in Ljubljana. First is complementary funding, and I'm looking at the Ministry of Science and Education here, which has written a very detailed and very legally binding commitment letter in the proposal. So this is something that will be checked in detail, and I trust it will be delivered given the positive experience with other teaming projects in the country. The second issue is international recruitment. I have already seen the proposals that we are targeting at least 20 full-time researchers based on international advertising. So this is something that will be looked at as well during the following time. Autonomy, also noticed in the proposal, in the description of action now in the project document, that the intention is to establish independent private entity as actually done by other running teaming projects of Slovenia, and I think this is also working towards this autonomy, whether in terms of setting research agenda, administration, hiring personnel. This is very much welcome. And last but not least, the sustainability. As you will know, the grant will end in 7 years, and then the centers of excellence are expected to run by themselves, to stay on their own feet. We do not know yet how does this work in practice, because now we are closing basically the teaming project resulting from the first call for 2016-17. And in this regard, as you will know, there is a teaming club meeting in Isola on the 28th and 29th of September. I trust you have already registered this issue of sustainability, how teaming projects, centers of excellence, in fact, will perform once EU grant is closed, will be discussed in depth there. One more thing to mention on feedback to policy. We will require every now and then for you to fill in a number of questionnaires, which will go very often beyond ticking some boxes. This is not just to do item list. It is something that we do use in our feedback to policymaker, which is DGRTD in the European Commission, because as you may have noticed, the widening program and the teaming action has undergone a number of significant changes from the first call until now. And these changes are actually based on this bilateral dialogue between the parties to the grant agreement. So I think this will be it for the moment. I will talk a hell of more tomorrow. Thanks again for the warm welcome in Ljubljana, warm welcomes, and looking forward to hearing from the project partners, if I got correctly from the agenda. Thanks again.
Sometimes this space is referred to, at least in my projection, as the Slovenian equivalent of what is in Finland called the Finnish sauna. But it's better now. Professor Zver actually mentioned it. It's better, the window is open, so you are also free to take your jacket off. Thank you very much for very kind words. Mislim, da zelo veliko povzemati ne rabim, bom vsem eno anekdoto povedal. V Romuniji, mislim, da je gospod iz Romunije, če se ne motim, pa se opravičujem, če ni, sem kolegico vprašal, kako vidi Slovence, pa je res lepo povedala. Rekla, vi ste eksperti zaprti v take male škatlice. Skratka, za nas je izziv sodelovanja. Mislim, da je tukaj lepo povedano, da pravzaprav po teh 7 letih mora center najti stik z še različnimi javnostmi, ki bodo omogočili preživetje. Without, again, to wait for the time, because we are half an hour actually too early. We shall continue with the next part, because there should be this natural flow. I would say nothing wrong with actually finishing with the opening remarks early. Professor Jarala, beseda je vaša.
Lepo pozdravljeni, gospod državni sekretar, direktor, spoštovani gostje, dragi prijatelji in kolegi. Hundreds of thousands of structures, of proteins, which are really the driving workhorses of living systems, but even more the application of machine learning to protein structure prediction that only a couple of years old really made huge leap forward, because now we have structures or reliable models of hundreds of millions of proteins, even more. Now we have the tools to modify, to design new proteins, those that have not been really found in nature and that have some very exciting potentials for therapies, and that's also one of the fields that we're active. All those tools and combined with many others are then used in synthetic biology, which is also a new discipline, I would say, a couple of decades old, that is enabling to introduce engineering principles into the biological systems. And now the reason for this project is that we can use those tools to translate them into the personalized medicine. One example, or not example, one field is of course to treat the underlying causes of disease with gene therapy, gene replacement, genome editing, and the second one is to use cells, engineered cells that act as a living medicine, and those are really more and more applied for therapy, and they are really the target of this call. Pharmaceutical therapy used to be limited to the small molecules, and there was a big evolution of biological drugs, and now the third pillar is being established based on gene and cell therapy that is becoming more and more powerful and targeted for individual patients, and we can address now diseases that have never been possible to be addressed before. So let's look at our expertise at National Institute of Chemistry. So I can say that we really cover the whole range from atoms to the whole to molecules. So we have the expertise and instrumentation as well. Cell biology to analyze, manipulate cells, but also deliver different drugs, such as using lipid nanoparticles. And also we have the in-house, in vivo experimental, I would say excellent, small, but quite sophisticated facility, which enables us to test this before going to patients, and to use it on different disease models, and to analyze the effect of those therapies. We can also say that one of the reasons is that we had some recognition for excellence for 2 advanced ERC grants in this area, and also one so far proof of the concept projects. And we have developed several diverse technologies in synthetic biology that could be potentially translated for clinics. I won't go into details in each of them, but there are different tools that enable modular sort of engineering principle modification, improvement of the efficiency of activation of certain genes, faster response than used to be with the technologies focus. Remember, we can achieve response within minutes rather than hours as transcription regulation. We can regulate cellular properties based on humanized proteins that are regulated by clinically approved drugs, which then avoid the problems of any potential immunogenicity. We also engineered modification improvement of CRISPR technology that enables us now to better, to have higher efficiency of genome disruption or targeted insertion. So in the area, the 2 main areas of our interest, as you heard already, are cancer immunotherapy. So the one where we are collaborating particularly with Professor Zver and Professor Sever at the Department of Hematology. We want to implement our tools or new tools into the improvement of cancer cell therapy based on CAR T cells to improve the efficiency and safety of therapy, to increase the indication for different types of cancer, and to introduce non-viral, more targeted introduction into the cell genome in order to have more efficient types of therapies. In the area of gene therapy, this really started off with engagement with Professor Sredka, who I'm happy to be here, and Špela, who really encouraged us to delve into this area. So we're using the CTNNB1 syndrome as a case study to develop new types of therapies for this particular, but also for other types of gene therapy. So this is the consortium that has been organized around the CCNNB1 Foundation, aimed to prepare a replacement gene, but also other alternative types of. What is really required is this triangle of collaboration between basic biomedical researchers, between clinicians, and between the interest of patients and patients' organization. Without of having the complete, so we realized that we should do something about, and rather to wait from the lotto to win, we decided that we should try to find out where are the possible sources, and we identified this teaming action as the only possibility to really get funding for this type of institution. So I'll briefly just review the last part of my presentation, how this started. So it really started basically slightly more than 2 years ago, in May 2021, when the concept was really crystallized that we should apply for this project. And then at this step, Professor Jurgen Kuball, who unfortunately cannot be today with us, then proposed the collaboration with UCL, which is already provided to be extremely fruitful. And that's what I'd like to thank to the people who really contributed to the success of this proposal. So people at the National Institute of Chemistry, I couldn't list all of them, but those that are most important, also with support of the leadership from the Institute of Chemistry, the Ministry of Science Education, our clinical partners, and also patient organizations. And I'd like to thank you for your attention.
The centers we sit in. The most tricky part I find always, and we have also patient representative here, so actually I'm curious about the chats you had, and we have the same, I would say, challenge in the Netherlands. At which stage and to what part involve the patients? Did you have any thoughts about this? Because I'm a hematologist by training, so we have a very vulnerable patient population. They have all different skills. What's your approach?
I mean, patients are involved in many ways. In the first place, they're the driving force, the motivation to do this. But in just the formal, we include the patients in the leadership of this organization. We involve patients' organization in some committees. After all, as we heard, the amount of funding to develop each therapy is quite big. So there are big, important decisions, and of course, at this stage, besides the scientists, where we can tell what's feasible or not, there will be also clinicians that will tell from their view, and also patient organizations what's really the challenges, the goals. They will help us set the goals, because we can tell honestly, we cannot promise to fill huge number of different diseases, but the goal is really to develop platforms that could be applied not just for a single disease, but the same next time for next disease. It should be much cheaper and much faster.
And maybe to the moderator, and also would not you only ask from the center here, but also from the expert part.
I have my mic. Thank you. Yes, thank you very much. And this question really puts us in front, I would say. So patient organizations in Slovenia have been here since 60s, 70s, 80s. They were never connected into one umbrella association. My interest in patient organizations came from the fact that I was interested in digital health since 2013. And my first invitation to patient organizations to participate in a digital health event, which I organized, was in 2016. I realized how much they are not up to what we expect using iPhones and internet, etc. Actually, still today, in many extents, when I invite patients to a conference we have next year, next Thursday, many of the patients don't respond to the question, please apply via this Google form. It's an anathema to them. So it's also even a further anathema, so not a topic to discuss the science behind the medicines. But you will always find certain very progressive patients, such as Speller, who was mentioned here, basically creating a genetic medicine for her son. And you will, in the patients, you will always find there is an argument we had before where the patients are always uneducated. Not at all, because all of us are patients at a certain point of time. With the patients, it's very important that you find organizations that is very, I would say, pristine, transparent, and driven by the goal, which is not, let's say, money or a position or something like that, but which is driven by a certain need, usually coming from the families, coming from the relatives. That's an important point. How to distinguish this? So it's important that these patient associations or patient organizations are true to their nature. Let's put it like that. And that comes through trust. You can't fake trust in the long run. You can put it in small bouts, pretend that you're a dolphin, and put on your fins and try to swim, but you're not a dolphin. And that's the point. You have to find the right ones. Professor Zver touched upon that a little bit. We have to find just partners who are willing to participate. In that sense, patient organizations are the best partners you can get. I can vouch for that, I'm sure. Thank you. Please.
I believe that, patients are kind of kings in our kingdom. If we consider most doctors being within. And I believe that they must know about themselves equal like we know about them. Because they're the one who will decide whether to follow our instructions or not. And in order to decide, they must know everything. So sometimes it will take you 10 minutes to achieve this level. On another occasion, 3 hours. Doesn't matter. We have to talk, and we have to respect them, and we have, as doctors, we have to consider the fact that they are those who will decide. And speaking of these therapies, about, let's say, cards are excellent, have excellent learning kind of curves. Let's say put elementary school cards produced by Big Pharma. Then you have such miltennis cards. These are kind of gymnasium. What Roman will do is medical faculty cards with academical, pure academical note. So it's an uprising learning curve from big pharma cards to products Chemical Institute will deliver.
Okay, I suggest we continue. Predlagam, da nadaljujemo. Dr. Mojca Benčina, v marsičem še danes omenjena kot ena od glavnih pogovorov tega projekta. Veseli me, da lahko predam besedo. Mojca, prosim.
Okay, thank you for the introduction, and thank you for you all coming here and celebrate the start of the very hard task, which is actually ahead of us. So, as you're probably aware, and I'm aware even more, it will not be easy. There are several reasons why, but nevertheless, we can start with the first part, what is actually the mission of the center. We have to be clear that the mission of the center is to develop gene and cell therapy, that's the mission, I mean the technologies for that, and that's for targeting neurological disease and immunotherapy of the cancer. But we have to also to prepare these innovative medicines for use in clinics in Slovenia. So that's the mission of the center. But of course, I mean, the goal is to provide Slovenian patients and clinicians with this access. So with the access to modern, effective treatment with the increase their availability. It means they should be cheaper as it is at the moment. So that's the main goal of the center, and of course that's not only goal of the center. I mean, we can in a line when we are going to that goal at the end, we can also benefit on other parts, and the other parts are to development of new technologies, which will not be only for our Slovenian patients, but it will be available for all the patients in the world. And of course, bringing biomedicine research directly to the patients and training the researchers as well. I mean, that's also an important part, to train our young, new coming researchers to the field of a new era of development of the drugs, because this is the era which will really expand and require more and more knowledge. So that's also an important part. Encouraging the creation of biotech startups. If you will be listening to the talk of our advanced partners, they will really stress that that's something which we need to consider as well. And of course, which we are already doing, bring the researchers, clinicians, and the patients together, so that we really go into the direction of the common needs. So what it will be the organization, because it's important. The founder of the center, it's National Institute of Chemistry, but all will be development and functionalizing the center will be bring together with our advanced partners, and it needs to be mentioned them several times, because we can't do this development without them, because they have a vast knowledge of their developing of the centers, and they will really try to persuade us that we don't teach, that we don't learn on our mistakes, but that they are actually learning without the mistakes they made before. So they are really crucial for us, and that's the University College of London, then the Center Charité Hospital from Berlin, University Hospital of Berlin, Utrecht University Medical Center, and Technical University of Technology from Dresden. So what are, we've already heard what we have. We've got money for that, so thank you for EU and also for our government. And there are 15 million euros provided by the two agencies, English and EU. And there are mainly funds for the working staff, so the employment, and then the 15 million from the cohesion funds, from our government, so to really build up the center and equip the center to really do the work. Where it will be stationed next to the National Institute of Chemistry, and will provide working space, the labs, equipment, and of course the knowledge. So that's something which is important. So for that, what will be the benefits? Because that's important. The benefits will be scientific, so we will be learning during the course, and we will also develop the technologies. Then there is a benefit of health for the patients, so we will be providing new technologies, easier accessible technologies, but there are also economical benefits for not only for Slovenia, but also to the closer region, and social benefits. At the end, we are actually going to a new direction, so with the gene therapies and immunotherapies, so it's important that also the wider audience really understand what we want to achieve, and also in this, they will also help us. So it's important that we all work together towards the common goal to get to the best, let's say, transfer and translation of knowledge to end products, so to get the patients benefit. So with this, I would like to thank you and give the word to the next speaker.
Thank you very much. I just wanted to start off by saying how delighted I am to be here finally after what seemed quite a long time from the initial discussions 2 years ago. And I'd just like to thank Roman and Mojca and the wider team here for your perseverance over those 2 years, and the hard work that you've put in to achieve one of these grants is amazing, and you should be congratulated on the efforts that have been put in. Of course, now the hard work continues. I also wanted to thank you for your patience for bearing with University College London as a partner, because at some times it seemed we would never get here because the UK is not necessarily everyone's favourite cup of tea, but I'm delighted that our government pulled through and gave us the funding for this project. So it's a really exciting opportunity, and UCL, I just put this slide up just to say we are a large, comprehensive university, very extensive, over 11,000 staff, 16,000 staff. So of that, over 3,000 are academics in the life and mental science area, with over 6,000 postgraduate researchers. So it's a big effort, and we have a lot of academics to work with. And in particular in this space, there's something like over 250 academics working in cell and gene therapies. So we've heard a lot about the ecosystem. We're very fortunate. Our ethos is to put the patient first, and so we've developed a long-standing relationships with a number of hospital partners. I think there's about 9 in our wider community, and I've picked out 3 here. Which is Great Ormond Street Hospital. So that's linked to our Institute of Child Health. So we go from the early stages of life right through to UCLH, which is a comprehensive medical hospital that covers all ages and all disease areas. And then Moorfields Eye Hospital, very focused, as it says in the title, on eyes and eye disease. The ecosystem. This is really, really important. The philosophy at UCL about putting the patient first, it really resonated with Roman's vision around what he wanted to achieve here and the setup. But what's really important is really understanding your local and your national ecosystem, from your hospital's clinicians, from who's really investing in you, from funders, equally from the national funders, international funders, charities, also investors like the VC community. Understanding your local environment, that's important for us in London, that we're addressing local needs and our local population of over 6 million patients. But also the links to government and the understanding of policy and what that can really do. You really have to pay attention to that early on, work together, really understand how they can support your motivation and how you can support theirs. So this is where our philosophy of health with wealth really helps in that. And the UK government has really invested heavily in cell and gene therapies. We have the cell and gene therapy catapult, which is aiding spin-out companies to really address that, one of the first hurdles in manufacturing. We've invested heavily, the MRC and LifeArc have invested heavily in manufacturing capabilities for the academic base. So that's sort of influencing that policy is really important. You've done a great job already in terms of convincing the government here to invest, and I think that's a terrific ambition that you've brought into. Industry, really important partners. Translation is, I say it all the time, is a team sport, and no one single organisation can achieve what we all want to achieve, which is impact in the society that we live in, impact for our patients. And industry, some academics still view them as a bit of a dirty word, but actually in order to to progress products to the market. They are a really valuable partner because they do share their knowledge and know-how and help you overcome those later stages in terms of getting marketing approval through. But first and foremost, patients and public. So I'm really delighted that you've got patient representation right at the heart of the centre, the proposed centre, because it's incredible. Unless you understand their particular needs, you're going to be developing things that perhaps aren't so important for the patients to be addressed. So really important to see them at the centre of everything. Okay, this is my last slide. So as I said, UCL is a large organisation with a large number of researchers in this field. The Translational Research Office, I joined UCL in 2009 and we started with three people. We're a team, we're a large team now of 17 people. And really we're out there trying to identify new projects, talk to the research base to understand their aims and ambitions and exactly how far they have taken their technology, and really help them navigate the complicated ecosystem within UCL, but also make partnerships with external organisations to help them progress their science. So in those first early steps, we've got some mechanisms to really help incentivise academics to work with us and also to identify those first early projects. And we call that, we have some early career research funding, which is about £10,000. That's to aid the early career researchers in getting their first grant and progressing along the translational pathway. And we see this as a talent pipeline. So this is an incentive for us to identify the talent that's out there and help them along that pathway and nurture that. This then feeds into our pilot data scheme, and that's about £75,000. It's about enough money for 1 year's project to take a TRL level 2 stage project to maybe a TRL level 3 or 4. And this is really important funding in order for us to build the product pipeline. And then that allows us to begin to work with those people to help them understand the development path. And then it helps us untap public funding. So the figures here are all for the cell and gene therapy pipeline. So to date, we've managed to help academics raise over £115 million in public funding. 6.7 million of that has come from UCL businesses area, but we advise on IP very early on. So we have a whole host of translational education programs here at this early stage so they understand some of the hurdles that they're going to face. We don't want to put them off, so we try and introduce them to that gently, but it's really important that you know where you want to go and some of the hurdles you're going to face. You make the right choices early on. So, how's this pipeline developed over the last 14 years? So we currently have 14 spin-outs in the cell and gene therapy area. Some of them are listed here on the right-hand side. We've licensed. You shouldn't forget the opportunity to license your technology to an industry partner. And this is a particular point in case. In 2013, we licensed a technology to BioMarine for haemophilia, and that's recently, last few months, got approval by the FDA. It's called Roctavian. We will begin to see some final...
They are just doctors, I observed actually the difference. So please, Professor, do.
Also, in line with the question about freshers, mostly it is the situation that we learn during the development phase many uncertainties to solve or to detect and to solve. And sometimes it is a very risky project we are dealing with because we cannot expect what will be happening with the patient. We cannot expect the side effects, neither the short nor the long lasting. and how you communicate this with the physicians, and most importantly with the VC, because the most important point is that VC, at least in Europe, is not very willing to go in higher risk projects and have a good feeling for this kind of treasures. So we at the end earn.
There's a couple of things in there. I'll start with VC investors. Just like companies, if you've dealt with an Novartis and a Pfizer, Alnylam, they are different beasts, all of them. That's the same with VCs. And for VCs, I think it's around, for them, they know what they're looking for and how it's going to fit their portfolio. So when you're communicating a risky project for VC investment, you probably have to go around and, as the phrase goes, kiss a lot of frogs, because you don't really know what the rest of their investment portfolio looks like and what their appetite for risk is. But generally, they're pretty risk averse, in my opinion. At the moment, it's a really bad time for VC investment in this space. It's in terms of funders, they have different motivations. So in the TRO, when we're looking for gems, we base that on unmet needs. Our tech transfer office, on the other hand, they have to really look with a hard commercial lens. So we might support something and see it through a much more charitable funded route. We also work on a joint portfolio, which is a little bit more commercial. In my experience, I think the ways of risk-taking just change. I mean, all it takes is for one bad example in the press, and then that's the whole, that's the whole field put back a little bit further. But on the other side, we're seeing some amazing advances, and the potential of these is really being pushed by patients, and that's why they're so important.
We have a question in the audience. Please introduce yourself.
So, GP Shuttle from UCL as well. Two comments, they are not a question. The first one is about your question and the fact that it deserves a very long discussion. And I think that we need really to bring to the VCs and the funders the concept of bidirectional research, that it goes so from discovery to patient, but then we needed to take it from patient back to the bench, because clearly we have one element of uncertainty that may progress. I'm working in the neurological space, so we are roughly 20 years behind cancer, and I think that we should really look at cancer, how the things evolve with the better discoveries done in cancer, with the toxicity but done in cancer, and so on. But I would like to add to the presentation of Jane by providing an additional bonus that the UCL can provide, which is the diversity. So I think that we in the UK have invested quite a lot in diversity of patient, in diversity of a collection of genetic data. And we can really bring the added bonus of diversity to Slovenia. Because I think that one of the problems of the unplanned discoveries is when we go to the patient, is the diversity of the patient that we treat. And I think that we may have done progress in that direction, that we can bring to the consortium and to TIVI.
Thank you very much. I suggest we continue with the next speaker. This is Professor Dr. Jurgen Kuball from the University Medical Center in Utrecht. If I need to correct my pronunciation, please do so.
Because you said access is very important, and I'm active since more than 10 years in the EBMT, first in the board, now I'm chair of the Legal and Regulatory Affairs Committee, where access of advanced therapies is one of our major goals. And when I saw the request of you, to be honest, I quickly checked your science, which was excellent, and the country, and this was a perfect match. So I'm really happy that you invited us. And the third one is a very simple one. When I just had food, I had a Wiener Schnitzel. And this reminded me of how close our cultural connection is. Yeah? And I think these are what these grants are about in Europe, in particular in these difficult times, that we remind us where we are coming from and that we connect and try to really work together. So I will talk about the cell and gel therapy programs at the UMC Utrecht. I have a couple of relevant conflicts of interest. You can see also on our webpage, and you will see also later. I have for sure too many slides, but luckily some of the speakers were not too long, so I will steal 5 minutes if I'm allowed. And first I will go on our research focus. And 15 years ago, when I left the United States, I was really thinking what's the best thing I could do as a young postdoc when I went to the Netherlands. And I started just out of the blue because I was impressed by some papers coming out about gamma delta T cells, that I will work about gamma delta T cells, a cell even most immunologists do not know very well. Luckily for me, this was a good educated guess, because nearly 10 years later there came a paper out where they looked at the clinical outcome of patients and the number of tumor-infiltrating cells, and to the biggest surprise of everybody, not the alpha beta T cells, which were the kickoff for all the checkpoint inhibitor treatment, were the number one, but the gamma delta T cells. And the most frequently studied, and in each conference you go, the NK cells are actually very bad T cells. So, and a lot of other papers came out confirming really the important role of gamma delta T cells in cancer immunotherapy. Doesn't say that the gamma delta T cell is the only one, but really a cell I would like to propagate, and luckily I know in London there are also very prominent gamma delta T cell people who are in the field. The gamma delta T cell even became more prominent in the area of CAR T, because what happened is that people transduced alpha beta T cells, that's what they thought. They had this famous first responding patients, and they looked then 10 years later what are the long-term surviving cells in these patients who apparently might keep in check the leukemia, and by accident they transduced a gamma delta T cell wearing a CAR. Wow. Then people said, yeah, but that's an accident. And the London colleagues, I think, had a second cohort where they looked also in their cohort, what is the long-term persisting cell in these patients. And the story is a little bit more complicated because they showed the first wave of alpha beta T cells, but also here they found long-term persisting gamma delta T cells, making the point that these cells may be totally underappreciated in a sleeping beauty moment, and I think is now really discovered from the community. Within our research team, so it's sort of a deal package you get with me, I just put the 5 prominent researchers in there. The one I'm really happy to join, and maybe Jort can stand up so that everybody can see Jort. Yeah, too shy to stand up or wave at everybody. Sebastian, who's one of the person of the first hours, who's working on prediction models. I will show a little bit of this later. We have Trudy Stratemans and Colin Deha, who's working on ATMP development. We have people working on clinical trials, and we are working also a lot of bispecific formats, because the ATMPs and bispecifics are, in my perception, also from the clinical perspective, very close, and you need to deal with both. Meaning, we are working on intellectual properties, writing patents about new leads, which is an urgent medical need for solid cancer. We are a lot of thinking about how do we engineer receptors, and I saw already a lot of interesting techniques where we might create unique win-wins with our unique receptors, but also I think unique techniques you have. We have really went through extensive immuno-oncology modeling. How can we predict that the one or the other choice is the best choice? Because we know as researchers we have too many choices in what is going to the clinic. And we made really the bridge from having the idea in the lab, making the manufacturing in the lab, bringing it to a full-grade GMP environment as you're planning here. And I can tell you we made many mistakes. Yeah, and I hope that we can tell you a little bit about our mistakes. And I learned also that the London colleagues made similar mistakes. So if mistakes are happening twice, don't do it the third time, it doesn't benefit. And that's why I'm very happy. And I think we had really also good publication output, the fun for the scientists. Some education about gamma delta T cells. If you never heard it, there is one important message, that there are really two main subpopulations. And we'll mainly talk about the delta 2 positive. So if you go into immunology, do research, never look only at gamma delta T cells alone. You need to look at the delta 2 positive and the delta 2 negative. That's really like CD4 and CD8 cells or NK cells and T cells. They're complete different biology. I will focus in this talk only on the delta 2 positive. And the idea is, and we had a couple of papers on this, that you have molecules which are called butyferylins, BTN2 and BTN3, which are mainly in a healthy state scattered around in the cell. And what is happening if a malignant transformation is happening, then with a small GTPase, row B, there were Scholt had a nice paper out, this is coming to the cell membrane, and it's like you're swimming in the ocean, the shark is biting, the arms come up, and this is seen by the gamma delta T cell receptor. How exactly this looks like was not very well known, and we have been lucky with our models, I will explain later, to be part of a paper which is just out in Nature, where they show that these molecules interact closely and come together, and like this, with a certain transcription program, this process is only active in the cancer cell, but not in healthy cells. So I think this riddle is nearly solved. And I know there's another paper in Nature in Review where they show even the binding crystal structure to even furthermore solve this issue. And this was for us a long time with talking to investors an issue, because people didn't exactly know how this works. We showed here, in this case, in breast cancer organoids, because that are the organoids we are working a lot in Utrecht, that this mode of action can be modulated with certain chemical compounds. And then if you look at different clinical cohorts, an example, low-grade glioma, you see if the gamma delta T cells are there, if the signature is there, then the patients have a better outcome. In line with the first paper I showed, but a little bit more sophisticated. Making again the point that gamma delta T cells might be very important at cancer for cancer immunotherapy, and we really need to dive into that. And if you just think about the diversity of these cells, you could put one cell next to the other, go from the Netherlands to Slovenia, around to the Netherlands, Slovenia again, and then maybe to, I don't know, halfway to developments. That's why we have been supported and lobbied in the Dutch government a lot. They call them national growth funds. We have been lucky to be part of the first one, which is about regenerative medicine, but has also an ATP facility. We got a structural grant, which is under the lead in Utrecht from Trudy Stratemans, one of our team members from the KWF, that's the Dutch Cancer Society, where we unite all the ATP facilities in the Netherlands about standard operating procedures, things nobody would like to share. And I can tell you it's still a very emotional process, so it's not that everybody is sharing now, but we're in the process of sharing. We got this last year another grant, which is also about viral production, and with a lot of innovation with companies who develop new devices for viral production, which might be also interesting for the year. And I think the biggest one we got is Oncode Accelerator, which will be officially launched, so I'm not officially allowed to tell, but I think in 4 or 5 days, which is a 400 million euro program from the Netherlands, where 40 million euros or 60 million euros goes to cell and gene therapy for the development of these infrastructures we need there. So another reason why I thought it's a perfect win-win to team up. And of course, Slovenia was the next one. The other one I cannot announce, but I can tell you it will be also very interesting for here, because this will be, has to do something with artificial intelligence and how to deal with CAR-T. If we go to the next one, so what is then important? So this one needs lead detection machinery, that's in black, very smart from me. And then the next one is lead prediction machinery, and that's where really Scholt comes in, with a paper he made with his colleague Florian Deckers, where we looked at breast cancer organoids. And the idea was to take our tech concept and try to understand how these techs work with solid cancer. And then you can do a lot of co-culture preparations, 3D light imaging, image processing, and single-cell transcriptomic. And the smart thing is you can image up to 150,000 cells, and you do multi-omics, if I say also what Schultz says, and then combine this also with the single-cell sequencing data and try to understand who of all these cells you're transfusing are the good cells, which are the bad cells, which cells might be not as good but still very important. And the findings were quite amazing because, like in whole life, you have all states. Some are dying, unfortunately, some are static, others are very lazy, others are slow scanners, high scanners tickle, they just come to the cell, tickle and go away, and others are really engagers and super engagers. And the question is, which composition of this product you need? So we infuse CAR-T and think that's one product, but that's not true. That's a million diversity of a product. And the product changes also between the patients. And really understanding how this works is important. And just one glimpse how we dive into this, and I hope this works now. So you see in the yellow, it's a breast cancer organoids, 20-30 organoids from a patient. The purple one, the bluish one is the engineered cell. And then if you look at this one cell, it's docking to these whole tumors, and this is about hours, and it's then spreading its tentacles around this tumor and is slowly biting into the tumor, and as it slowly gets red, is really eating up the whole tumor mass. And then the understanding should be, what is the property of this cell? And then you can do from the attached cells single-cell sequencing, and then you find a lot of new genes, which are in this case about morphological plasticity. You're not surprised if you see this picture, which you really say this is an important property, and that's just the tip of the iceberg we're looking at. Another paper now nearly accepted, hopefully, in Nature Immunology, where we show how we can tip the balance. Yeah, that goes to the one or the other right. I think this is exactly maybe also tools which might be very interesting depending on the choices you are doing. This is a picture of the different cells in this example. So we have the text, that's the concept I described. We have a WT1 TCR, which is not the London, but the Seattle WT1 TCR, and a CAR-T, and just compare the profiles. And the nutshell is, CAR-T frequently dies, we know this, but it's very hyperactive. The TCR is a slow scanner, but also super engager sometimes. And you need to tip the balance somewhere in between. Yeah, and finding this and having all the choices, therefore I said also CD90 CAR-T is nice, but that's just the tip of the iceberg. If you would like to be innovative, you need to be somewhere in the middle, and we have a lot of tools to make it in the middle. Potential future here, for example, thinking about CAR-Gs, where gamma deltas are carriers, but I mean, this can go in every direction we are thinking of. The most important one is then ATMP optimization. How do you make your drug? And I think the colleagues from London, ourselves, we have similar approaches where we have biofabrication, because it's linked to regenerative medicine, and also the GMP simulation facility under the leadership of Trudy Stratemans, and the GMP production facility under the leadership of Colin Bauer. And the idea, the center part of this, is a GMP simulation facility, which hopefully makes it quicker to go from the lab to the clinic. And the question is, what is this? So basically, it's the exact same equipment as in the GMP clean room. You have there the interface between researchers and the specialists and other experts, because they're completely different, speaking two different languages. If a researcher thinks I just take another medium because the other one is not available, it's a major change from a GMP perspective. Yeah, and these different worlds need to be connected. And we believe that this efficiently allows the translation from clinic to the clinic, and I heard from the London colleagues they made the same experience. And you can also do all the documentation at a way much lower price, because you do not need to pay the clinic price. There we provide then trained and qualified personnel, and I think that's also an idea that might be interesting to link to the idea here. We have special tech transfer processes. For example, we are participating, maybe you've seen a paper about tumor-infiltrating lymphocytes from our colleagues from John Hahnen for melanoma patients, which is getting into ensured treatment for patients, where we participate in the production process. We develop a lot of analytical methods, and of course the GMP compliant documentation. We have all these folders of documentation just to make one product. And then the other people in the clinic ask, why does it take so long? Just because they don't understand. And I think really bringing this understanding to the clinicians, to also your authorities, and I was really thrilled that so many authorities were showing up here, because you really need to have also some stamina to go through this route, but it's really worthwhile. Because it really makes a change. I mean, in the Netherlands, just as a clinical example, CAR-T was approved by EMA, but not paid by the insurance companies. And it has a 30% survival benefit long-term for patients. So basically, 20 to 30% of Dutch patients died because they didn't have access. And I think, to my best knowledge, the same is happening also here. Because you don't have access, you lose 20-30% of young patients, of young ALL patients, which could be otherwise easily treated. So therefore, I'm even more happy that this is happening. So take-home message is gamma delta T cells are the new black in cancer therapy. That's not from me, that's from this review, but I think it's really interesting. Understanding cellular receptor diversity is key. Targeting innate immune receptors is really interesting, but the challenge is in safety and efficacy testing. So we have to have smart models like mouse models, 3D models. And I think it's worth a challenge. We saw a complete response of an idea at 15 years ago, just now, but you really need to work and the speed on the technology transfer. Brings me to thanking a whole team, Scholt, who's here, Trudy, Dennis, Monique de Witte is another one, and of course a whole team working on this stuff. Thank you for having us and inviting us.
Okay. I'll go back a little bit more to the organizational part, what we learned and what we have, let's say, established at the Charité to build up an ecosystem of cell and gene therapy. So, I will start with what was already mentioned and discussed. And this is that we as a clinician have to face, we have to face the situation that in Europe more than 30% are patients affected by chronic diseases. And this is related to the diseases, some of them are already mentioned. One is cancer, of course. Academic institutions are more prominent in comparison to industries. One point is quite clear, it was mentioned by Jurgen. Manufacturing is a crucial point. How you manufacture and how you characterize. Mostly developed by our own. Yeah, so, and I can tell you, the first product we developed, this is not only a matter of time, it's also a matter of documentation. And we had for the first, only one, which is not related to all renewals you have to do, it was 5-kilometer paperwork. And only for getting an approval for the blood. This is the source we are working with, not leukapheresis, also, but we focus on blood. It took us 3 years to get the approval, and it took us, this is only 13% of our whole production process. And we end up with a height of documentation like the Eiffel Tower. Yeah? So to get an impression, what does it mean at the end to start with a clinical trial? So, our government, in particular our Wissenschaftsrat, Council of Research, we can translate this, was fine with our concept. We get 30 million for building up a new GMP facility, which is around 10 clean rooms. And the intention of our center is early phase IITs, speed up, of course, transdisciplinary, this is what also was mentioned, and transdisciplinary, I will come back shortly, to develop innovative technology and in a scalable perspective. to develop manufacturing standards and self-product analysis, and this means we have to cooperate with everybody. And this is a cartoon showing you what we mean with, let's say, transdisciplinarity. At the end, very slow, we have a discovery team, we have an enabling and technology development team, and we have the clinician. And what we have established is a joint project team dedicated to each product which is of interest for a dedicated indication. And we have defined, which is also not easy, I have to say, different go and no-go checkpoints at different stages towards the clinical trial. And it was also mentioned here the aspect of the defined translation to have this kind of iterative improvement. So because if you come up with a new ATMP, whatever it is, for whatever the indication is, academia take over mostly the whole pipeline part, and we can do it on an academic GMP production, on a hospital GMP production, or we can go to a CMO and take all processes over, make something easier, but it is not the case. But at the end, we want to have the product for the patient, for all patients in need available. And here we have, let's say, two ways. One is to focus on near patient and stage production, which is the strategy of some companies. Milton is one example. Or you have what the big pharmas are looking for, off-the-shelf delivery of products. So, with it in mind, we started several years ago together with Johann van Elderen. This is a UHA, let's say, consortium. This is a European Hospital Alliance, in which these universities around Europe are interacting and close connected, and so. And within this European Hospital Alliance, we built up the UCAT as a virtual European institute, again to transfer manufacturing technologies among us, and to develop products together to advance clinical stage. And, thanks to the European Commission, this, let's say, UCAT consortium get now the funding for CSA within the Horizon Europe. And I see it because it was a long way. It was not easy to achieve it. And we are leading this joint ATMP. And I think to not only to say how good we are, but to convince you that we are able to set the right points also regarding networking. Because networking is everybody speaking about, but in most cases it is not really a network, because everybody is claiming their own interest and looking only, let's say, to have the money for their own interest. But for us, for me, networking is more. It's really to build up an infrastructure globally, at least in Europe, and focusing, of course, on different medical needs and aspects. And this is what we have established. We are leading, and here we are, luckily, I have to say, really luckily, partner in this kind of widening program. And I can promise that we do our best to help with our knowledge. This is very brief what our expertise are. Our expertise is related to enhanced targeting of cell samples. It means redirecting of specificity, homing aspects play an important role. Do the cells really home to the targets they have to go? And the lasting intertissue functionality, because mostly the cells come in an unfriendly environment, which means in inflamed area. It is even the case in cancer. So that you have at least a local, mostly also a systemic inflammation, which target the cells you prepare outside. The second point is, of course, the viral vector gene editing or vector-free. We are focusing, as you know, you visit us already, more or less on the non-viral vector gene editing strategies. How you enrich the cells, this was topic of Jurgen already. Yeah, and you can be sure that you never get a pure product. Yeah, this will not be, but you have to be as sure as possible. And this is also technology related. That your sorting strategy is really convincing you that you have the majority of the right cells you want to have in your product in place. Expansion strategy and of course single cell analysis, and Jurgen already mentioned the importance of preclinical models, and here I think organoids are already mentioned, and human organ on the ship is also important. So this is more or less my last slide, because I think this is also a message towards the European Commissioner or Commission. For me it is not only an honor and a privilege to be a partner, it is also one of the best dissemination strategies we can have, because in all EU resorts you have a huge part of dissemination. Yeah? So, but this is indeed dissemination, because we have to setting up GMP facility, we help in innovation, yeah? We provide contact with the regulatory authorities, which is very helpful. You can make a lot of mistakes and turns with them. I think we have, you more than we, experience with industry and VC, which is of importance with respect to the sustainability. Startups and training are already mentioned. So that's what I have to say shortly, more or less. Thank you.
Thank you very much. I apologize that I'm not there. I'm actually on vacation. It's my only week, and I've not had a single day of vacation while I'm on vacation. But so it's fine. I think we're all used to that. And so a little bit about me while I share, if I'm allowed, grant access in the system. Allow WebEx to share. Okay. Good. Oh my God. Here we go. Sorry about this. Let's see. Okay. Data. Let me see. It won't let me open. Why is that? That's what I'll do. I'll look at my slides and tell you all about them. So just a little bit about me. I'm Ezio Bonifacio. You can tell that by my accent that I'm actually originally Australian. I'm not German, but I have an Italian background. And I actually was in Milan at the San Raffaele Institute, which I'm sure you all know very well, for 13 years or so, some time ago, and I was director of their pilot transplantation program. Why I have a strong interest in cell therapy, and of course partnered the Telecom Institute of Gene Therapy, so interested in gene editing. And I'm in Dresden. In Dresden, we are at the Centre for Regenerative Therapies, and we in Dresden have a few years ago come together with our colleagues in Leipzig, and this is a venture that in Germany is called a Cluster for Future. It is a venture which funds a joint partnership between academia and industry in order to develop industry in a region. And of course, the region is Saxony that we're trying to develop, and the area is cell and gene therapy. And Ulrike Kohl, who's at the Fraunhofer Institute for Cell Therapy, is my co-speaker in this venture. I think it's an important venture because it's very much dealing with some of the things that we have been talking, or you've been talking about today, and we will be doing over the next years, and that is to move things not just scientifically, but towards a therapy for patients, but also industry. And that's a real challenge, as you know, because scientists don't think that way, or at least the ones I know. So it's a real culture change for many people. As I said, it's a partnership between Dresden, Leipzig, and also Chemnitz, and a large number also of industry. I think I don't really want to go too much into the areas, except to say, obviously, the areas of focus for us are the CAR-Ts, the CAR-NKs, but also other cell therapies, including macrophages. We have a program that is trying, like many people, look at universal cells and also modulating therapies. I did want to speak a little bit about the platforms that we have, or some of our core areas in Dresden, where we think we can contribute to this consortium based on where they've gone so far. One of them is the area of gene editing or genome editing, and you know, for us it's not all CRISPR. We have Frank Buchholz as a major player in Dresden. Frank Buchholz is designer recombinases. That's what he's researched for more than 2 decades, and is really an expert in that, and has brought that to therapeutic levels where it's very predictable and precise, so it's very, very safe. It takes longer than the CRISPR, but it really can address almost any target. You can excise unwanted DNA, for example, HIV, which patients have in them, which he can remove, and you can insert into safe harbors wherever you decide. You can invert, for example, with factor VIII, and you can exchange any mutation. So this is a technology that is built around Frank Buchholz's core. He's also spun out companies, Prohirix and more recently Seamless Therapeutics. And so it really is an example of taking something from the science right through to company and also therapy. The other area that is important for us, where I think we made some important contribution, has been in the what was originally called the UniCAR models, so modular therapy, where there is an adapter molecule that is the versatile part of it. It's controllable. Where the CAR T is at least half of the matter, the adapter molecule is what activates it, targets it. And that is in clinical trials. Of course, there was a startup, Kemijski, which is now Avencell, sitting in Dresden. So these are a couple of our areas that we're happy to, or we sort of proud of and are looking forward to contribute. Through Saxicell, we've learned how to cooperate among competing places. That's actually never as easy as you'd like, but it has been a learning process. And we've also created a harbour, a place of coming together, where we have tasks which we do together, which is to manage and communicate with the individual projects so that they are moving towards their goal, looking for venture capital, looking for that outreach wherever that's required, both at the industry side, but also public and government side. So this is done, I think many places from the talks I've heard have this, and I think this would be a great share opportunity amongst us, because it is an important part of what we're all trying to do. We also have some platforms, we call them, clinical trials for the omics, which is the monitoring, and for systems, which is very much the automated processing of cell production. Some of the novel areas that we have, I'm looking at my last slide at the moment, so some of the more novel areas is, of course, that our vision, like yours, go beyond CAR T cells, rare diseases and cancer, and we do look at new cells. We have Mojca Benčina, who is with us and has renewable sources of macrophages, macrophage cell therapy, where these are gene edited macrophages, and is taking that to a setup. We have Anke Fuchs, who works more on the immune dampening side of things with other cells, Tregs, for example, etc., and some other interesting ideas. So we're very, very happy to be part of this. Very happy to be with such excellent partners, and to
introduction. I will conclude in English on behalf of our international guests. Thank you also, organizers, for inviting me and give me the opportunity to say something about patient perspective or expectation from this kind of centers and therapies. I earlier saw your disclosures, Jurgen, you showed. I always put them, but my biggest bias and disclosure is that I'm a patient myself. Exactly 15 years ago today, I was diagnosed with breast cancer. Now I know that that is really an early age. I was 26 at that time. So at that point, I just wished that I will live until I'll be 40, and that by that time I will throw a party. So I did. Last year, because of the COVID, I was 40 during the COVID, but last year, my husband and me, we had a big party for our 40th birthday. It was actually a concert. Gregor was also invited. So celebrate life, live, survive. Do we want just to survive or do we want to live? My second disclosure is that I'm a scientist, or I used to be a scientist. I have a PhD in molecular neuroscience. Another Gregor was my professor during my PhD. And another Gregor, which happened to be a rector today, was my mentor. So I quit science. Did I really? No, I didn't. I still am a scientist. I still love science. I'm still part of many steering committees of research projects. But my profession now is a patient advocate, patient representative. Using all my knowledge that I got during my trainings to make this environment better. Maybe we can thank European Commission and EMA that they put it in their actual guidelines that there is really nothing about us without us. Earlier, Gregor also mentioned that at some point everybody is a patient, even if we're not aware either we are using a healthcare system. But today maybe you feel healthy, the next day you can get a diagnosis of cancer or any other rare disease. In that case, definitely research, development, and everything regarding the new technologies is very welcome. At the beginning, a few times it was already mentioned how patients are important and that we are in the center. But actually, during these past years, we learned that we actually don't want to be just in the center of the circle, but we want to be a part of it. Being involved in research from the very beginning, being involved in policy making from the very beginning, because at the end, you are the one who are receiving the treatment. And also development of the treatment, how it went from one size fit all to more tailored, personalized, individualized treatment, whatever you want. But this is nothing possible without science and research that I can live with them. You cannot see that I have more than 80% physical damage on my body, and then officially I'm a disabled person. So, but this is something that you learn to live with, and that quality of life is something that you can survive and live this life. So that's all from my side, and I really wish this center to become successful and that we see the first patient get the treatment from this center in the next at least 10 years, and that this will become a reality. No pressure. You already said that you are a big pressure. Thank you.
Another one of her talents, and not in the language of the boy, I mean of language, but she was dancing on the floor. So she used to be a dancer. She wanted to be a dancer. Now she's a patient advocate. She knows a lot about science. So thank you for accepting the suggestion. And of course, without diminishing the role of other patient organizations and everybody in the triangle that was mentioned here. And in this regard, please allow me to use what Dr. First said earlier when we discussed. We need to drive, I hope that we are capable of directing the development of what we are doing with the acknowledgement of the differences that are enriching us, and also that not all things are to be just described in words. Some need to be danced, some need to be felt, some are there in spirit or in the goodwill. This applies a little bit to ethics, it applies to culture, it applies to who we are as a society and what is the meaning of what we are doing here. I am very convinced that the meaning and the biggest celebrations in life are not those that have a big, nice, fat check behind them. We are aware that in order to do this, we do need the support of the European Commission, the state, and everybody else. Thank you very much. This was a wonderful kickoff from my perspective, and I'm really grateful for having the opportunity to be your moderator today. Upam, da vas nisem za komentarji pravzaprav mogoče zapeljal v smeri, ki ne bi bila zaželena. Kakor sem povedal, hvala, da ste tudi ostali. Hvala, da bomo tudi v prihodnje lahko sodelovali.